Hello everyone, welcome back to my channel. This is a full walkthrough, answer and explanation for October 2024 Unit 3, and please subscribe if you want more parts and if you also would like a final Unit 3 revision video. Okay, so the first question is, maize is grown as a source of starch. The photograph shows a picture of a ripe maize corn. Okay, cob, I mean. Describe the structure of starch. Okay, so this is a repeated question that is repeated in multiple exams. So starch is composed of alpha glucose. Remember, we have alpha glucose and beta glucose. So starch here is made up of alpha glucose molecules that are joined by 1,4 glycosidic bonds. So starch is also composed of amylose, which is coiled, and amylopectin, which is also branched. Now, they also might ask you a question, what is the significance of starch being composed or branched? You can say that it will store more energy, and when it's branched, it's easier to break down, so energy is released quicker. This is also a possible thing they can ask you. Okay, now question B. In maize, starch is synthesized at an optimum temperature between 20 degrees and 30. Okay, so the first keyword is optimum temperature. When I read optimum temperature, the first thing I thought about was enzymes. Okay, so the question wants us to explain the effect this will have on the mass of starch produced in maize plants. Okay, so we have a temperature between 20 and 30. So I'm going to choose a sweet spot, which is going to be 25. So my first point is most starch is synthesized at 25 degrees Celsius. Okay, this is probably the optimum temperature because 30 might be too high and 20 might be too low. And then we're going to say since starch is made using enzymes. Okay, and then these will give you the 2 marks. Question II. Studies have shown the effect of temperature on starch synthesis. In an investigation, the rate of starch synthesis was measured at 4 different temperatures. The results are shown in the graph. So on our y-axis, we have the mean rate of starch synthesis, milligrams per day, and then we have temperature on our x-axis. So the next question is going to ask us to make a table of this data, and it's going to ask us to look for the standard deviation value at 30 degrees Celsius. So since I can't go back to a slide, I'm just going to explain it right over here. Okay, so to find the standard deviation, all you have to do is count the units on this side or on this side. When I counted them, it's 0.35. Okay, so they're going to ask you over here to draw a table to show the results from the graph, excluding the standard deviations. Okay, so obviously you're going to have to draw this out in pencil, and a mistake over here that I did, which is very difficult to fix using a computer, but in pen and pencil you can. You have to erase this out. Okay, these things over here, you must erase them, and it must be like a neat box. Okay, obviously using a ruler, and then for your values you're going to be using a pen. Okay, explain what the value for the standard deviation at 30 degrees Celsius means in this investigation. So in the previous slide, I showed you how I got this value of 0.35. Obviously, never forget to write down the units because the units alone is 1 mark. So now let's explain what standard deviation is. So standard deviation is a measure of the variation of data around the mean. It's very important to write down that it's around the mean. Okay, the mean is the average value. Question IV. Justify conclusions about the changes in mean rate of starch synthesis between 20 degrees Celsius and 25 degrees Celsius and between 25 and 30 degrees Celsius. Okay, so between 20 and 25, when you're answering these questions, make sure to always be looking at the graph. So the difference between 20 degrees and 25 degrees Celsius is significant since standard deviations do not overlap. When error bars or standard deviations do not overlap, there is statistical significance. Okay, However, between 25 and 30, it's not significant because the error bars do overlap, okay? So that means that in the range of values of both temperatures, there is a value that is the same. Okay, question C. The graph shows the synthesis of starch at 30 degrees over a period of 16 minutes. The table shows the initial rates of reactions calculated for 4 other temperatures. Calculate the initial rate of reaction for 30 degrees Celsius shown in the graph. Okay, so focus on the initial part over here. They said initial, so therefore I'm going to be focusing on this point. I am not going to be taking this one and this one. No, I'm only going to be taking this point over here. So all you have to do is our simple equation, which is y2 minus y1 over x2 minus x1. This is our gradient or slope equation. Since our y1 and x1 are both 0, all we're going to do is 16 divided by 4, which gives us 4. Therefore, our answer is 4 mg per minute. Plot a graph of the data in the table and join the points with straight lines. Okay, obviously your points over here are going to be in x's. Do not do a dot and do not make a star. Just do a simple x. And then draw your straight lines with pencil. Your titles are going to be in pen, and also make sure to cover more than 50% of the graph. So question triple I. Give 2 reasons why the initial rate of reaction was measured. Okay, so we measure the initial rate in any experiment that we do so that it allows for the comparison of rates between temperatures. If you didn't measure the initial rate, then how will you make a comparison? Okay, it doesn't make sense. Point number 2 is the initial rate would not be influenced by a change in substrate concentration. So this means that the change in concentration will not affect the initial rate. Okay, question number 2. More farmers are using environmentally friendly fertilizers. So just when you're reading this question, one thing that you can point out is usually environmentally friendly fertilizers are really expensive to make. So one example is vermicompost, okay, which is VCL. It's a liquid formed when worms decompose organic matter. So just think about how many worms the farmer would need to just have a simple dot of fertilizer. Whole gland solution contains the mineral ions required for plant growth. 3 mineral deficient solutions were made from whole gland solution. Solution had no nitrates, so no nitrogen. Remember, nitrogen is needed for protein synthesis. Solution B had no phosphate. Solution C had no potassium. The effect on the dry mass and length of shoots of tomato plants treated with these mineral deficient solutions supplemented with VCL was investigated. The photograph shows a picture or shows a tomato plant. So we have our shoot and our roots. The graph shows details of the treatments and the results for shoot dry mass. Over here we have the complete whole glands. The black one is with VCL, the white one is without, okay? And then with no nitrogen, no phosphorus, no potassium. Notice how with the potassium and with the phosphorus, with the VCL gave a higher amount of dry mass. With the no nitrogen, it's almost the same level. However, I can't see if it's exactly the same, but it looks about the same, and it's very low. Also with the complete whole glands, you can see that there is more growth with the VCL. Okay, describe a method that could be used to obtain the results for the complete whole glands treatment so that it can be compared with the other treatments. So number 1 is we're going to be using a water culture. Okay, because using a water culture, you can easily control the amounts of nutrients. If you use soil, it's very difficult to control the amount of nutrients present in the soil and the pH. So we're going to be using a water culture. So a water culture basically is growing plants in water. So I don't know if you ever seen the vertical farm. The vertical farm is like a tub. There's a tub over here, and then we have like holes, and then the plants grow out of these holes. So like usually it's like lettuce or basil, and it's just a bunch of holes. And this over here on the base, it contains water, and the water travels up. This is one of the ideas of the water culture, or also you can have multiple cups of water and you can call it a water culture. So one culture will contain the VCL and one is going to be without the VCL. Now our control variable is the type of tomato plant used. So we're going to have to be using the same type of tomato plant. Do not use species, okay? Because multiple different type of tomato plants can belong to the same species, okay? The temperature and light intensity must also be controlled so that it's a fair growing environment. And then we're going to remove the roots and dry to then find the mass, okay? Calculate the percentage increase in shoot dry mass with VCL when VCL is added to Hoagland's solution. Okay, so if you look at the previous graph, I labeled 115 and 95. So we're going to get 115 minus 95, which gives us 20. Now 20 over 95 times 100 is going to give us 21%. Question BI. Suggest why shoot dry mass is a more accurate measure than the length of a fresh shoot. Okay, so first of all, when you have a plant shoot, it's going to be very difficult to say where the roots begin. So some might say that the roots begin over here. Some might say they begin over here. Some might say they begin over here. Okay, and also a fresh shoot will contain water, which will lead to a variability of mass of tissue. Okay, the photograph shows the appearance of plants following each treatment. The photograph for each pair of plants has its own scale bar. We have the one that's not done for us is the no potassium with VCL. Complete the table to show the actual length between X and Y for the tomato plant labeled no K with VCL. So over here we're looking for the actual shoot length. Okay, so when you measure using a ruler this part, this is not your actual. You're going to need to use the scale bar. Okay, and you're also going to need to be measuring the scale bar over here. When you measure the scale bar, it's going to be 5 using your ruler. So from X to Y, it's going to be 38 millimeters. Okay, then we're going to multiply that by 50 divided by 5, which is going to give us 380, and just write your value over here. Discuss the use of VCL as a fertilizer. So VCL obviously enhances the growth when both potassium and phosphorus are absent. Also, VCL has very small effects on growth when when nitrates is absent. VCL improves root growth in all treatments. Okay, if you see in the graph, you can see that with VCL, it's slightly higher than all of them without. However, VCL fertilizer may be too expensive to use. Remember how I talked about how it might be difficult to gather many worms and collect only small amounts? Also, there are no error bars present in the graph to show statistical significance. So yes, the results might seem different. However, we don't know because there's no error bars to further provide the statistical evidence needed. Okay, question number 3. Petri dishes with nutrient agar were used in an investigation of the antimicrobial properties of plant extracts. This is one of the required practicals that is repeated in multiple exams. So it's very important that you understand the steps. Bacteria were spread on the surface of the agar to form a lawn. Plant extracts were placed in wells cut in the agar. After incubation, the size of the zone of inhibition is measured of the antimicrobial effect of the plant extract. The diagram shows a petri dish after the incubation period. Remember that there needs to be sufficient time for the incubation to occur and it needs to be at sufficient temperature. because these are microbes that need different different growth properties to allow it to grow, and also we need to give enough time for the plant extract to work, because if you take medication, it's not going to work the second you swallow the pill. It's going to take maybe after 4 hours for the effect to start happening. Okay, so explain why the dishes were incubated for 48 hours at 30 degrees Celsius. So 48 hours allows time for the extract to work. You can also say it allows for the microbes, sorry, to start growing. And at 30 degrees, it will not allow the growth of human pathogens, okay? So this 30 degrees, it might be sufficient for the microbes. However, it will not allow growth for different pathogens from the outside of the environment, which can possibly or do actually contaminate the agar plate. Explain 3 aseptic techniques that should be used when transferring bacteria from one culture to another. So remember, we're going to have to spread the bacteria using a loop. So a loop looks like this, and it's a stick with a circle in the middle where you can use to spread around the agar plate. So number 1 is we're going to flame the loop. So we're going to put this loop under a flame from the Bunsen burner. Okay, this kills the bacteria on it, so all we can say is flame the loop to sterilize it. Point number 2 is you're going to want to work near a lit Bunsen burner to move microorganisms in the air away. When you have a Bunsen burner lit up, there is going to be convection. So convection is the heat is going to move like this. The hot air is going to rise and fall, and it's going to be moving away the organisms. It just looks like this. Point number 3 obviously is use ethanol on workspace to kill bacteria. You can say use ethanol or use methanol or just use any alcohol to wipe the surface. Question B. The technique described was used to investigate the antimicrobial effects of oils, so of oil A and oil B. The oil A and B is extracted from 2 species of plants. The bacteria that we have is made of E. coli and B. subtilis. Explain how the area of the zone of inhibition could be measured accurately. So number 1 is obviously we're going to be measuring the diameter using calipers. So this just means is that we're going to measure it from every single angle. However, just memorize calipers as it's easier than writing from every single angle. And then we're going to be calculating the area. Since this is a circle, our calculation for the area is pi r squared. This is repeated and this is also repeated. So memorize these 2 points and understand them. Okay, state one independent variable in this investigation. So the independent variable is what you are in charge of. So in this investigation, you're in charge of the type of extract, oil A and oil B, and you're also in charge of the type of E. coli or the type of B. subtilis that you want to use. The table shows the results from this investigation. So we have our type of bacteria, B and E, we have oil A and oil B, and we have our zone of inhibition. So the larger the zone of inhibition, the more effective, because that means that this zone has no growth on it. Determine the effect of these 2 oils on the growth of these bacteria. So since we have a table with values given, it's very important that you manipulate these values and you give them some math, okay? Because showing your use of the data and using one mathematical point or statistical point is going to give you 1 mark. So I decided that I'm going to divide 20.66 by 11.33 to give us the difference, which is going to be 1.82. Therefore, oil A has an inhibition zone that is 1.82 times than B. Oil A is a more effective inhibitor of bacterial growth than oil B. Okay, since its inhibition zone is larger. Okay, now both oils are more effective on B. subtilis. If you can see over here, the oils are more effective on B. subtilis. The inhibition zone for B. subtilis is fairly larger than for E. coli. The smallest antimicrobial effect is oil B on E. coli, where it gave us 0. So absolutely nothing happened. Now question C. The size of the zones of inhibition may also be affected by the rate of diffusion of the antimicrobial molecules through the agar. Suggest two factors that may affect the rate of diffusion of the antimicrobial molecules. Number one is incubation temperature. Number two is the size of the antimicrobial property. Okay, now our final question. A method that avoids this problem is to find the minimum inhibitory concentration, MIC, of the extract. The MIC can be defined as the minimum concentration of the antimicrobial molecule that will reduce the number of living bacterial cells by 90%. Okay, meaning that we have 10% left. The next question is going to ask us to find this 10%. In an investigation, the data were obtained. So basically, I'm going to find out what 10% of is 200, which is 10% of 200 is 20. So the number of living bacterial cells is going to be 20. I'm going to look over here at 20 and find the concentration, which is going to be 4.3. And here's the calculation. Thank you so much for listening to my video, and I hope that it was helpful. Please subscribe and comment if this video was helpful for more videos in the future. Thank you so much.